Okay. Good afternoon, everyone. My name is Shripriya Kalbhavi, and I'm from Lynbrook High School in San Jose, California. I'm very thankful for the opportunity to be able to present my research today, and this was conducted mainly on identifying a novel method of active medication delivery via microneedles using a Belousov-Zhabotinsky oscillating hydrogel. So, in addition to my research on this active medication delivery system, I specifically wanted to identify potential sustainable and affordable drug delivery systems. So, to give an overview of what I'll be talking about today, I'll be starting with a brief introduction on my topic, and then I'll be going into a brief literary review of the different works that I looked at previously. And then I'm going to be talking about mainly my research procedures, then my results and findings, and I'll be wrapping up with some discussions and further considerations on my topic. So, to give a brief introduction, the hypodermic needle and syringe was identified by Dr. Alexander Wood in 1853. The hypodermic needle was an extremely innovative solution to drug delivery because at the time there were no other real main methods that could help with effective medication delivery during medical procedures. So this was extremely revolutionary at the time. However, since 1853, the initial design of these hypodermic needles has not changed much, and therefore these hypodermic needles remain a source of fear, pain, and discomfort for the end users, as 2 in 3 children and 1 in 4 adults report a severe needle phobia. So this severe needle phobia affects many people, not only in the United States, but all over the world. And even it can cause other severe side effects along with being afraid of needles for many people. Because, for instance, during the COVID-19 pandemic, up to 1 in 10 people in the United States postponed getting a COVID-19 vaccination due to their fear of needles. So, as we can see from this, not only does these hypodermic needles cause fear and pain in many people in the United States and around the world, but it can also lead to other adverse consequences, such as putting off vaccination as a whole. This is extremely negative because during their first year alone, especially, COVID-19 vaccination saved around 19.2 million lives. So, because a lot of people put off the vaccination, a lot more lives could have been saved. Also, older individuals, as well as patients with medical conditions such as forms of dementia or Alzheimer's disease, face difficulty in taking medications, especially taking medications autonomously. They might also forget to take their medications. So these are all problems that could arise with being an older individual who needs to take these different medications. So therefore, the current gaps in the market include the absence of painless, autonomous medication delivery systems. This is something that I wanted to look into and identify a potential solution for. There are also some other alternative solutions in the market today. For example, there are passive drug delivery systems, which essentially do not have the active force that hypodermic needles and other forms of medication delivery, like swallowing pills, do. These passive drug delivery systems, though they are not painful, they do have other limitations, including having to rely on natural diffusion for the drug delivery. So this limits them by systems such as the 500 Dalton rule, which means that generally medications that are over 500 Daltons in molecular weight cannot be transported into the skin and therefore the bloodstream via transdermal patches. This led me to finding a new type of solution for this problem, which is through microneedle arrays. These were first characterized in a paper in 1998 by Dr. Mark Prausnitz at Georgia Tech. Microneedles are generally 150 to 1,500 micrometers in height. They're hundreds of times smaller than the traditional hypodermic needle, and they are much less painful than these hypodermic needles because they're reduced in size. And overall, people generally describe it as feeling like sandpaper or a cat's tongue or something like that. There are many different evolving technologies of these microneedles, including dissolving microneedles, coated, hollow, polymer, hydrogel, etc. In my research, I specifically looked at hollow microneedles for delivering medication through my patch and into the skin and bloodstream. So, when these microneedles are combined with an active form of medication delivery, they can autonomously deliver medication into the skin. And this is specifically, I studied this through the Belousov-Zhabotinsky oscillating reaction. This fluctuates with the concentration of bromide ions in solution when the iron catalyst, specifically phenanthrylene ferrous sulfate, shifts between its 2+ and 3+ state. So this not only changes the color of the reaction, but it also changes overall the composition. And research is currently being done on how this reaction could be controlled via lasers, electrical currents, etc. But specifically, what interested me about the BZ reaction is the fact that it can be incorporated into highly soluble hydrogels that are extremely susceptible to environmental changes, such as temperature, pH, etc. And these hydrogels can swell and shrink with respect to the concentration and cycles oscillations. So what I wanted to do was address the issues of painless and autonomous active medication delivery by incorporating a Belousov-Zhabotinsky oscillating hydrogel into a microneedle patch. So I just conducted a brief literary review. Mainly I looked into previous problems in the drug delivery industry. And on top of that, I did some research into the BZ hydrogels and how they are being synthesized by various researchers. The first BZ hydrogel was discovered by Ryo Yoshida in 1996, but after that there have been other papers that have gone into the synthesis of this reaction. And specifically, I looked into two main papers to refer to for my synthesis, which was Jiren et al. 2017, and Takashi Arimura and Masaru Mukai et al. 2014. So my initial procedure consisted of synthesizing two types of gel in order to analyze the various microgel, not only the microgel but also the homogeneous gel's particle sizes. What I was aiming to discover was that through the variation of these particle sizes, they would be able to generate more pressure with larger particle sizes, because this would be able to take in more water and be highly swellable. So my first procedure was the microgel synthesis, utilizing Jiren et al. 2017. My main procedure for this involved using the prepolymer anisopropyl acrylamide. I also had another copolymer in this case, which is acrylic acid, and my catalyst was 1,10-phenanthrylene 5-acrylamide. And then my crosslinker was BIS crosslinker, and then I used ammonium persulfate as my initiator. So I would be conducting an emulsification polymerization over here. And lastly, I used SDS in order to lubricate the reaction. So my initial emulsification procedure, I was basically able to perform an oil bath and add in the reactants and the ammonium persulfate. Once this was complete, I ran my microgels through the rotovap, and I was only able to yield a very small amount. So this procedure is not highly efficient, but I was able to synthesize microgels using this procedure. I was able to dissolve my residue in ethanol on DCM, and once it was done, I left it to evaporate in a fume hood. So these are my synthesized microgels after polymerization and recovery, and as you can see, it's not a particularly large amount, so this was one of the downsides of this synthesis, but I was able to obtain microgels that I could use in my experimental procedure. And over here you can also see the chemical structure of these oscillating microgels. You can see that the ferroin group that I was trying to attach overall to the hydrogel is over here. It has been polymerized. So this was my main goal for my primary procedure using the microgels. However, I not only wanted to synthesize microgels, but I also wanted to synthesize gels with a larger particle size. So specifically, I ended up synthesizing a homogeneous solid gel. I actually synthesized about three different types of solid gels, but I used a different catalyst as well to see if I would be able to polymerize the catalyst and see if that would make any particularly huge difference when I was looking into the oscillation pressures that these gels generated, and would be able to help better deliver medication into the bloodstream via the microneedle patch. So the procedure overall was quite similar to the previous microgel synthesis procedure. However, there is one pretty big differentiator. I'll talk about that in 1 second, but similarly to the previous procedure, I used N-isopropyl acrylamide, I used BIS crosslinker, and I actually used a few different catalysts, but the one that ended up being the most effective was iron 5-acrylamide 1,10-phenanthrylene BIS 1,10-phenanthrylene sulfate. My main difference between this procedure for the solid gel versus the microgels procedure was the utilization of AIBN as my initiator instead of ammonium persulfate. And the AIBN is what differentiated it from the emulsification reaction versus the solid gel. So I performed a similar polymerization. Essentially, I used three main different catalysts. My first catalyst was 1,10-phenanthroline iron 2 sulfate. The problem with this catalyst was, even though it was somewhat effective, as you can see over here, a lot of the catalyst actually did not polymerize and combine with the gel like I'd wanted it to. Mainly because it was missing that acrylamide group that allowed it to attach to the rest of the hydrogel that was being synthesized. So this particular catalyst was not quite effective when I was performing later tests on it. Next, I had a second synthesized gel. I used 1,10-phenanthroline 5-acrylamide. This was somewhat closer to what I was trying to actually achieve for my overall structure. It had the acrylamide group, but also this was not particularly effective as I was missing one more key component in order to help it attach and polymerize. My final catalyst, which I synthesized, was iron 5-acrylamide 1,10-phenanthroline bis 1,10-phenanthroline sulfate. This catalyst actually ended up being the one that attached to the overall gel. This was the more effective one, and this one was able to generate the most pressure from the oscillations when I was performing my later tests on it. So I was also able to construct the microneedle patch. I made a plexiglass prototype, and I also made the actual patch, as you can see over here. My plexiglass prototype consisted of a few main key components, which was the piston, sink, hammock, medication reservoir, and a syringe with an attached microneedle tip. So to explain the general mechanics of how the patch works, so the patch has a sink in which the inactivated hydrogel would be sitting in. Once the gel is activated, essentially the piston would be pushing down on top to force the hydrogel to push downwards through the hammock onto the medication. So this would push down on the medication and force it through the microneedles and into the skin, and by diffusion into the bloodstream. This was made out of plexiglass. This was my initial prototype over here. And later, I also constructed a physical patch just to be able to model what this patch would actually work like in real time. So I also had another main highlight for this. I had a fluidic pressure measurement system. My fluidic pressure measurement system was basically used in my later tests in order to gain an understanding of how much pressure oscillations my gels were able to generate. This was their main purpose and usage. So I was able to construct this using a disposable pressure transducer, and this was linked with an Arduino system with an Arduino board, software, and breakout board and wires for probing. My pressure transducer over here, this disposable pressure transducer, compared to like actual pressure transducers they might use in a hospital setting, these ones are disposable, so they're much less expensive, and I'm also able to set up the system at a relatively low cost. So my pressure transducer was also able to generate readings in volts, so I had to use an additional Python program and function to convert this data from volts into mmHg. So my pressure measurement system was connected to the plexiglass prototype where the microneedles were being run through by the medication. You can see over here, this is the microneedle tip that was attached to the pressure transducer. So I also introduced a breakout board to increase the accuracy of the readings, and I calibrated the entire system using a manometer and aneroid sphygmomanometer that I'd taken apart to create baseline readings and to verify the accuracy and validity of the data. And overall, my hydrogels were characterized using BZ reactants and methylene blue. I also looked at them underneath the microscope. So to characterize my gels, I also first looked at it by adding BZ reactants to see their oscillations. I was able to identify a 7% oscillation, and I also observed these solid gels underneath the microscope, and you can see what it looks like over here. So I also was able to analyze my gels and characterize them through my plexiglass prototype. What I did was, into the sink of the plexiglass prototype, I was able to add my BZ gel along with the reactants. And then I used a methylene blue as my model drug. So I used my DAC, data acquisition unit, and pressure transducer setup to measure my my pressure measurement recordings. So I was able to find that the solid BZ gel had the most significant results. These were generating 8 mmHg of pressure and 0.625 centimeters of displacement, which you can see through the hammock over here. It was able to generate 0.625 centimeters. So overall, my results and findings found that my BZ reactants were able to, when combined with my solid gel that had the most effective catalyst, that was the last iron catalyst that I'd spoken about earlier. This was able to generate significant displacement of the hammock, as seen in the previous slide. The hammock was pushed significantly downwards. And this generated 0.625 centimeters in depth difference, and the pressure transducer and DAQ also detected an 8 mmHg difference in pressure generated by the hydrogels. This is extremely significant because of the low amount of hydrogel and the small size of the patch, so this should theoretically be able to provide enough pressure to move my medication through the microneedle patch and into the skin and bloodstream of the patient. So I also found that my microgels actually were not able to displace the copolyester hammock significantly, and this did not generate any significant pressure changes. The synthesis procedure was also extremely inefficient, so these were not very efficient in delivering medication. Over here you can see my calibration data for my pressure transducer. This was how I matched the voltage values from the pressure transducer with various mmHg values using the aneroid sphygmomanometer as well. This is my oscillation data from one cycle of the oscillating gel, and you can see the increase in 8 mmHg of pressure over here. So to wrap up my presentation with some conclusions and discussions, my research found that utilizing a homogeneous Belousov-Zhabotinsky oscillating hydrogel, which was initiated with AIBN, produces the best oscillating motions that generate pressure and force the active autonomous medication delivery in a microneedle patch. So I also found that utilizing my pressure transducer system, I was able to actively monitor readings and gather accurate data. And this was able to detect significant changes in pressure caused by the homogeneous oscillating gel versus the microgels. As for my results with the actual solid gel, I was able to generate 8 mmHg of pressure and displace the hammock by 0.625 centimeters. These were my most significant results because the microneedles and the other gels were not able to significantly displace the hammock or generate a significant amount of pressure. So this oscillating gel in the future, it could be potentially used to deliver medication through a microneedle patch, and this could be controlled via the dosage and intervals by potentially being pre-treated with external stimuli, such as lasers and electrical currents. Other types of oscillating reactions could potentially be explored in the future as well to be used in the system, and it could be used at a doctor's office when administering something as short-term as a vaccination, or it could even be used as a long-term solution for medication delivery at home for patients to use. What I want to look into in the future is exploring the reusability of the patch. Some things I was considering was adding a valve to the reservoir so medication could be reloaded. I could also make sure that there are steps taken to prevent the misuse of the patch. And I could also use customized syringes to deliver only certain amounts of medication or pre-filled medication packets, etc. So these were just some things I was considering. Lastly, just the scope of my work. I was able to design not only a new microneedle patch system, but also implement oscillating hydrogels. I received a provisional patent for this microneedle patch in February of 2024. And I was able to conduct my chemistry portion of my project at the Panorama Research Institute under Mr. Cyrus Becker, who was my mentor and also oversaw my procedures and made sure I was conducting them safely. So these are my supplemental information and my references. So thank you for the opportunity to present about my research today, and it's extremely incredible opportunity to be able to share my my research to many distinguished scientists today. So thank you for your time.